So in this question, we're going to talk about a molecule called dinitrophenol, or DNP. And DNP is a molecule that can diffuse into the intermembrane spaces of the mitochondria, and it can bind with protons, and it can carry the protons across the outer or the inner mitochondrial membrane. So instead of the protons going through the stalk particle from the intermembrane space back into the matrix by facilitated diffusion, they're being carried out of the intermembrane space. So the energy, instead of being used to generate ATP, is actually now being lost as heat. So the way of thinking about DNP as a molecule is, maybe imagine you trying to ride a bike and the chain came off, and then you had to cycle really fast, but actually nothing's really happening. The bike isn't going to move very much, and that's a bit like DNP. So as the electrochemical gradient is being built, the DNP molecule is actually diminishing or making smaller the electrochemical gradient because it's removing the protons from that intermembrane space. So we're going to annotate the question. It does say that DNP transports protons across biological membranes, including both mitochondrial membranes. It does decrease the proton gradient, so it diminishes the electrochemical gradient, and instead of being used to generate ATP, it's lost as heat. So the first question says, following establishment of an electrochemical gradient, describe the normal route that the protons would take in order to synthesize ATP. So we know there's an accumulation of protons in the intermembrane space of the mitochondrion, and we know they normally move by facilitated diffusion back into the matrix, and the energy that's being released from that movement is then used by ATP synthase to combine ADP and PI to make ATP, and that's part of oxidative phosphorylation. But the question underneath is asking us to explain how DNP acts as an insecticide. So an insecticide is any molecule that can cause the death of insects, which might be pests, for example, on cropland. So in order to act as an insecticide, DNP, because it's diminishing the electrochemical gradient, it means fewer molecules of ATP are being made by ATP synthase. So we can have the idea that not enough, a lower yield, or less, or even no ATP is being produced by oxidative phosphorylation, but we need to reference why it's causing death of the insects. So a lack of ATP in the cells, for example, the muscle cells, means muscle contraction wouldn't be able to occur, and therefore there'd be no flight of the insects. There'd be no cellular responses such as protein synthesis, active transport, transmission of nerve impulses down the neurons. However, it does say, please do not accept overheating or denaturing of the proteins in the insect. Now, if DNP is used in humans, it can cause denaturing of some of the proteins in the cells of the human, but because insects are so small, they're going to have a much bigger surface area to volume ratio compared to humans, and therefore they can dissipate the excess heat more efficiently, which might mean actually that some of the proteins or the enzymes are not necessarily being denatured. So the main problem in insects is the lack of ATP and the lack of the processes that would normally use ATP. So part C goes on to describe the use of DNP in the 1930s as a dieting drug. And it does tell us that after a number of years, DNP was identified as causing severe side effects, including death. So DNP was designated an extremely dangerous drug and not fit for human consumption, so its use was actually banned. Then it says more recently, DNP has again caused some deaths in people taking it to lose weight. And these people were people who might have obtained DNP, but maybe they used too much of it, maybe the dosage wasn't correct. So the first question says, explain how in detail DNP brings about the symptoms described. So it is used as a weight loss drug because if we think about the substrates for respiration, we know glucose is used in respiration, and if there's not enough glucose, we know the glycogen in the liver and the muscle can be hydrolyzed to release alpha glucose. But if the glucose can't meet the demands for ATP, then alternative substrates will get used. So the fat stores or the triglycerides in the liver cells of the insects, or the humans even in this case, will get used up. We know triglycerides can be hydrolyzed to fatty acids and glycerol, and the fatty acids can enter respiration and they'll get used in, it's called beta oxidation. So the use of fat stores or triglycerides or fatty acids as an energy source will cause the weight loss in addition to the respiration of glucose. Part B says increased metabolic rate to compensate for ATP underproduction. So what that means is you need a faster rate of respiration in order to try to regenerate the electrochemical gradient, because the DNP is continuously reducing that electrochemical gradient by removing the protons. So the rate of respiration will increase quite dramatically. It's like if you're on a bike and you're moving the pedals, you want to move the pedals faster because the bike's not really moving very much. And it's like the rate of respiration going up and up, even though the gradient is being diminished. Now, because of a lack of ATP, there's going to be the fatigue that was given in the information. So we know fatigue really means tiredness, and that means the muscles can't contract quite so easily. So that's why you feel the fatigue. And then the heat released as a byproduct of increased rates of respiration will increase the body temperature, probably above the core body temperature of 37 degrees C. So that's why it is very dangerous for humans, because they can start to overheat, and therefore some of the enzymes in the cells or the proteins in the cells could start to denature. Now, it does say in the information about increased levels of sweating. Now, if you go back to the water topic, we know that sweat is largely made from water, and water has a large latent heat of vaporization. So that means it can actually take away a lot of the heat energy from the blood in order to vaporize water or to evaporate water, to remove excess heat from the body. And then lastly, part D says, how might DNP cause the rapid death of a person who's taking it to lose weight? So we can have the idea of overheating, denaturing of the enzymes in the cells or the tissues. We could have some reference to organ failure due to a lack of enzyme activity or a lack of ATP production. And not hypothermia, but hyperthermia is another term used for overheating. So the next question is a basic recall question asking us to describe the reactions that link glycolysis in the cytoplasm to the Krebs cycle in the mitochondrial matrix. So we know pyruvate is the substrate for the link reaction in the mitochondrial matrix. So pyruvate can actually get into the matrix through both inner and outer mitochondrial membranes through carrier proteins. There we know it's decarboxylated, so it's oxidized. So the hydrogen is removed to make NADH or reduced NAD. That's by dehydrogenase again. We also know that the pyruvate gets decarboxylated, so a molecule of carbon dioxide is removed as the carbon atom is removed from pyruvate. And the 2 carbon that's left over is called acetate. And then we know the acetate combines with coenzyme A to form acetyl coenzyme A. So dehydrogenation is the same as oxidation, produces NADH. Decarboxylation removes CO2. By decarboxylase, the 2 carbon acetate combines with coenzyme A to make acetyl coenzyme A. Okay, glycolysis is in the cytoplasm of all cells, including bacteria, and the Krebs cycle will occur in the mitochondrial matrix inside, obviously, the mitochondria of eukaryotic cells. So the next question is about the Krebs cycle. And we know the Krebs cycle happens inside the mitochondria, in the matrix, the fluid organic matrix of the mitochondrion. And we know acetyl coenzyme A at the top is made from the link reaction. So we're just quickly going to annotate the diagram, and then we can answer some of the questions. So the 2 carbon acetate from the acetyl coenzyme A is then combined with the 4 carbon, and it says oxaloacetate on the left. And that's going to make the 6 carbon citric acid or citrate molecule that's just been highlighted in the red box. Now we know citrate, or the 6 carbon molecule, is then converted into a 5 carbon intermediate. Now we don't have to know the names of these intermediates, although it does often help us a lot. Now, because carbon has been lost, the atom of carbon has been lost as a molecule of carbon dioxide, this is going to be decarboxylation by decarboxylase. But also some hydrogen has been lost from citrate, so citrate has been oxidized, and the hydrogen has been converted or transferred, onto NAD to make NADH. So there's 2 products from this first step, step P. CO2 is made and NADH is also being made. Now the 5 carbon alpha ketoglutarate, the same thing happens there. So that is also oxidized to lose hydrogen onto NAD to make NADH, and it's also decarboxylated to release carbon dioxide. But this time there's an extra step that happens. So the energy released from the oxidation and decarboxylation is used actually to combine ADP and PI directly to make a molecule of ATP. So this is what we would call substrate level phosphorylation, where a molecule of ATP has been made, the high energy bond has been formed through condensation, and the energy has been released from the breakdown or the oxidation of the 5-carbon intermediate as it's being converted to the 4-carbon. Now the first 4-carbon is called succinate, and then succinate is converted to another 4-carbon called fumarate, or just a 4-carbon intermediate, okay. Now, because there's no change in the carbon number, there's no more loss of carbon as carbon dioxide. However, there is oxidation of succinate. So when it loses its hydrogen, it's transferred onto FAD this time, not NAD. So we're going to get a single molecule of FADH. Now, if we have a look at the fumarate, the 4-carbon intermediate at the bottom, there is an enzyme in the mitochondrial matrix called isomerase. So it's going to convert the fumarate into a slightly different structure, and this new 4-carbon intermediate is called malate. So there's no loss of hydrogen, there's no oxidation here, and there's no loss of carbon dioxide. But once we've got malate, that is then converted in the matrix to oxaloacetate at the top, which we know is another 4-carbon intermediate. But the malate actually does lose hydrogen, so it's being oxidized by dehydrogenase, and this time the coenzyme is going to be NAD, so we're going to get another molecule of NADH. So from one Krebs cycle, we've got 2 molecules of carbon dioxide, 3 molecules of NADH, and a single molecule of FADH. Now, an additional note that you might want to add onto this picture is that each NADH that's being made, that will carry the hydrogen to the start of the electron transport chain in the inner mitochondrial membrane. And we know NADH will become oxidized, so it will release its hydrogen, it loses hydrogen, which then splits into electrons and protons. The electrons move down the ETC through electron carriers. Energy is lost from the electrons. That activates the proton pumps, and it pumps the hydrogen ions into the intermembrane space. So every NADH will provide enough hydrogen ions and electrons to make 3 ATP by oxidative phosphorylation. So because we've got 3 NADH, that would produce 9 ATP indirectly through the establishment of the electrochemical gradient. Now, if you have a look at the FADH at the bottom, so this is step R, FADH, when it becomes oxidized, it loses hydrogen at the second proton pump, not the first. So the electrons move through the ETC from the second pump onwards. So that's going to activate only 2 proton pumps, which builds a slightly smaller electrochemical gradient, which is sufficient only to make 2 ATP by oxidative phosphorylation. So decarboxylation then, the enzyme is called decarboxylase, and it's the removal of a molecule of carbon dioxide from an existing intermediate. Dehydrogenation then is essentially oxidation, and it's the removal of hydrogen, which we transferred onto a coenzyme such as NAD or FAD. And the 2 letters that we just discussed were P and Q from the Krebs cycle. So the next part of the question is asking us to work out the number of ATP molecules that might be formed from either a single link reaction using NADH or a single Krebs cycle using the 3 NADH that are made from a single Krebs cycle, or it could be from one Krebs cycle using the single FADH that's made. So because 1 NADH is made in the link reaction, that's going to mean 3 ATP are made at the electron transport chain, ATP synthase in the stalk particle. 3 NADH are made from a single Krebs cycle, so we know 3 ATP are made per NADH, that's 9. And because a single FADH is made in the Krebs cycle, that activates 2 proton pumps in the inner mitochondrial membrane at the electron transport chain, so each FADH will make 2 ATP. And then the question at the bottom is saying, explain why the 2 hydrogen acceptors, NAD and FAD, lead to the production of different numbers of ATP. So a single NAD will provide the hydrogen ions and the electrons. The electrons go through the entire electron transport chain, activating 3 separate proton pumps. FADH, when that's oxidized, the electrons will only go through 2 proton pumps. So 2 proton pumps get activated as the electrons go down the ETC. So there's a bigger electrochemical gradient, or a steeper concentration gradient of hydrogen ions when NADH is oxidized compared to FADH. So a very common type of exam question in terms of practical work is the use of a respirometer. And a respirometer can be used to estimate respiration rates in different organisms, including yeast. So in the diagram, we have a glass flask, and there's a culture of yeast within the flask, and they're exposed to glucose, so they can use the glucose as the substrate for respiration. But there's also a liquid called pyrogallol, and it does tell us that pyrogallol absorbs oxygen gas from the air within the flask. So because there's no oxygen gas after, say, an hour, when you start the experiment, the yeast are forced to respire anaerobically. So we know yeast are facultative anaerobes, and they can respire in the absence of oxygen. Now, the two products at the end of the anaerobic respiration or alcoholic fermentation will be ethanol and carbon dioxide gas. So there's also a glass capillary tube that's connected to the flask. Now, this question is all about the pressure change within the flask. So if the yeast are respiring anaerobically and they're making carbon dioxide gas, but there's no oxygen gas, then there's going to be more gas molecules within the flask over time as the CO2 starts to accumulate. And that means the pressure in the flask goes up. Now, as the pressure increases, that's going to force the coloured liquid to move to the right. So we can actually measure in millimeters how far that coloured drop of coloured liquid moves. And then we can actually work out the volume of gas that's being produced through anaerobic respiration, because we can use the volume of a cylinder equation. For that, though, we would need to know the diameter of the glass capillary tube, which would give us the radius. So pi very often is 3.14. So 3.14 times the radius squared times the length that the coloured liquid did move in, say, for example, an hour, or it could be half an hour, or some time period that they give us. And that is the production of carbon dioxide gas from the yeast. Now, the first question says, why is it important that the yeast were left for 1 hour before taking the first measurement? Well, the pyrogallol, when you put it in a flask, it's not going to absorb oxygen directly straight away. It's going to take some time. So it's really important to give enough time, which they consider to be 1 hour, for the O2 gas to be fully absorbed to force the yeast to respire anaerobically. It's also very often to allow the yeast to have a respiration rate that stabilizes at that given temperature. So if you're going to do the experiment at 30 degrees C, then the yeast need to have a nice stable respiration rate at that temperature, which can take some time. So during her investigation, the coloured liquid moved to the right-hand side of the capillary tube. And just to quickly explain why it did that, well, we know the yeast respire anaerobically, they make carbon dioxide gas as well as some ethanol, and the accumulation of carbon dioxide gas, in addition to no oxygen being present in the air, increases the pressure inside the flask. Now, the maths question underneath gives us some values. So it says the coloured droplet moves 1.5 centimetres in 24 hours. Then it gives us the diameter of the lumen of the glass tubing, but it actually gives us that in millimetres. So because we're asking for centimetre cubed as a volume of gas per hour, we do need to convert the millimetres into centimetres. Now, the diameter of 1 millimetre is 0.1 centimetre, and therefore the radius is half the diameter, so the radius must be 0.05 centimetres. Now, if we then use the equation we're given, pi r squared times L, so we know 3.14 is pi, so please don't use the button on the calculator for that, just use what they give you. L is length, so 3.14 times 0.05 squared times 1.5, but because it says per hour, we need to divide by 24, because the original volume would be in 1 day, 24 hours, so if we divide by 24, we get 2 per hour. So always look at the units first whenever you do a maths question. So if you plug those values into the calculator, you should get 4.9, or you could have 4.91 times 10 to the power minus 4 centimetre cubed per minute, per hour.